Thank you. So this is my talk. I start with an overview. This is just to prime everybody on what is coming in the lectures which will ensue after my talk. Uh, so I'm going to talk broadly on immune organs, immune cells, effector immune molecules, and immune functions. Um, the immune organs include the primary lymphoid organs and the secondary lymphoid organs. Primary include thymus and bone marrow, and secondary ones include spleen and lymph nodes, as well as mucosa associated lymphoid tissue, which is present in the gastrointestinal tract as well as the respiratory airway epithelium and few other tissues. Thymus is a bilobed structure lying above the heart, and it is the capsular structure. It's lobulated with outer cortex and inner medulla. Uh, as you can see in this picture, uh, so under the capsule we have this trabeculae which divides the thymic tissue, and we have in the cortex we have the dividing thymocytes, the mature thymocytes, and some of the dead cells, and you also have some nurse cells which sort of nourish the thymocytes, and we have cortical epithelial cells and interdigitating dendritic cells also, and macrophages are also there, both in the cortex and medulla. In the medulla we have the macrophages, we have the Hassel's corpuscles, and we have interdigitating dendritic cells, and the thymus predominantly helps in maturation of T cells, enhancing the diversity of T cells and T cell selection. The T cell selection basically is based on the MHC antigen complex recognition, whether the T cells will recognize the MHC non-self antigen complexes, or it will recognize MHC self antigen complexes, or it does not recognize MHC antigen complexes. So these are very tricky balances to keep our immune defense system going on optimally without harming ourselves. You can know that the athymic conditions give rise to immunodeficiencies. You heard of de Georges syndrome, etc. T cell maturation happens around 8th or 9th week of gestation in humans. It has positive selection which permits the survival of only the those T cells whose TCRs are capable of recognizing self MHC molecules, and negative selection eliminates the T cells that react too strongly with the self MHC or with the self MHC plus self peptides. This is important, otherwise our immune system will be all the time fighting with our own body, and this is how it prevents autoimmune phenomenon to happen. At the same time, it also helps in fighting virus, bacteria, and other infection. Then comes bone marrow. In the microscopic structure, there are many things, so I will be mentioning a few, but the role of the stromal cells are important, and important functions of bone marrow include hematopoiesis, B cell maturation, B cell selection, and it puts out mature naive B cells into circulation. So this is an oversimplified picture of B cell development. You can see from stem cell, pro B cell, and pre B cell up to immature B cells. It is antigen independent process that happens in the bone marrow. Once it is exposed to antigen, then the immature B cells grow into mature and recirculating B cells, and germinal center B cells, memory B cells, and plasma cells in the spleen and lymph nodes, and they also recirculate back to the bone marrow in the red marrow. So the B cell development is happens, the initial part, naive part in the bone marrow, whereas more mature and memory and plasma cells happens in the peripheral tissue. So the same thing is shown in this picture here. The antigen independent process is from stem cells to pro B cell, pre B cell and immature B cell, whereas the antigen dependent process is when the immature B cells on antigenic exposure become naive B cell to naive activated B cell to germinal center B cells, post germinal center B cells, memory cells, plasma blast, which eventually become plasma cells and travel back to bone marrow in circulation. So this is a oversimplified history of B cell development. Now, secondary lymphoid organs include spleen, lymph nodes and the malt, as I mentioned. The lymph nodes grossly is a bean-shaped structure. It drains major segments of lymphoid tissue. Lymph nodes have microscopic structures which include major cell types like lymphocytes, macrophages and dendritic cells. It has got cortex, paracortex and medulla, and it has got primary and secondary follicles. So if you look at this picture, so this is how the lymph nodes and the lymphatic vessels are connected. And if you see the structure of the lymph node, so you can see that the cortex here in this light yellow part, and we have the blue part is the paracortical area, and the brownish or pinkish part is the medullary area. So in the cortex we have primary lymphoid follicles containing mostly B cells. We have also secondary lymphoid follicles with germinal centers. So this is how a germinal center mature one looks like, and this is a senescent dying germinal center. And in the paracortex we have mostly T cells, and in the medullary cord we have macrophages and plasma cells. Now from the capsule you can see we have the sinuses, we have the cortical sinus, we have the marginal sinus, and we also have the medullary sinus, and we have the artery and the vein, as well as efferent and the afferent lymphatic vessels. So this is how is the overall gross structure of lymph nodes. The same thing is shown here. You can see here that you have afferent lymphatic vessels, you have efferent lymphatic vessels, we have the lymphatic artery, lymphatic vein, we have post capillary venule, and under the capsule we have the post capillary venule has been expanded and shown here, and under the capsule we have germinal center, as you can see, with a lot of B lymphocytes here, and you have primary lymphoid follicles as it is shown here. This is the capsule. So this is a oversimplified picture of the uh, lymph nodes. So lymph nodes act as the first line response to antigens. Secondary follicles or germinal centers is the site of B cell proliferation and differentiation. It has got high specificity. More than 90% of B cells die through apoptosis. And after antigen stimulation, lymphocyte numbers go up by 50 times almost in the efferent lymphatic vessels when they go out, and lymphadenopathy happens in response to antigenic stimulation, whether it is a microbe or otherwise. Spleen, all of us know, it has a gastric surface, it has a renal surface and splenic artery vein, and this is the hilus through which the artery vein enters. The spleen also microscopically has red pulp and white pulp, as you can see here, white pulp and red pulp, and we also have the periarticular lymphoid sheath, a very important site which actually is responsible site for antigen presentation. The spleen filters out older RBCs, it responds to antigen in circulatory system and produces activated B cells. So we have the capsule, and we have the white pulp which also has the venous sinuses here. You can see the trabecular artery and the trabecular vein here. So on the red pulp is the background you can see, and if you see the transverse section and the longitudinal section of a white pulp, so you can see from perifollicular zone to the marginal zone to the germinal center and B cell corona. We have the red pulp outside, and we have the periarticular arteriolar lymphoid sheath and the central arteriole. So this is very important to remember, because spleen has two major of the two major regions. The white pulp or the lymphatic part, it has small arterioles surrounded by the pals, as I've told you, and it's surrounded by marginal zone. The red pulp clears RBCs, and it has cords of cells including erythrocytes, macrophages, dendritic cells, few lymphocytes, and plasma cells also contain sinusoids. The mole tissue, actually they are present on the mucosal surfaces of mouth, respiratory and reproductive tracts, and they are colonized by lymphocytes and accessory cells. It is also in the gastrointestinal tract. It responds to ingested or inhaled antigens. Also they are present in tonsils, Peyer's patches, and the appendix. So this is a picture of Peyer's patches of the GIT. If you under the muscular layer, there is submucosa with germinal center, primary follicle, and the lamina propria. There are M cells and there is follicle, and then we have the villi where the inductive sites, where the antigenic stimulus or the bacteria, virus infected cells can enter. The malt in Peyer's patches, actually if you see the same picture, the M cells are very important. Antigen is presented through these unique cells of the M cells. There is subepithelial dome with germinal center and T cells there. There are also B cells, and in addition to that, it is important to know, like in any mucosal tissue, when the antigen from the bacteria enters through the M cells in the lamina propria in the organized lymphoid follicle, they activate plasma cells, and the plasma cells produce IgA. As you know, the mucosal immune response is predominantly IgA driven. And there are also intraepithelial lymphocytes, you can see between the M cells here. The tonsils are follicular structures. It contains lymphocytes, macrophages, mast cells, germinal centers, and germinal centers appear in response to antigen, and they have protective role in upper respiratory infection. The tonsils are many, palatine and lingual tonsils are here, pharyngeal and tubal tonsil are there, and along with the nodes around it, all these tonsils together, this is called the Walde's ring, and you must be knowing the recent discovery of further lymphoid tissue in the pharynx, which was described probably a couple of months back. Appendix is also associated with the malt, and it responds to antigen, and it has got some role in GI immune response. The movement of lymphocytes is also important. The B cells from the bone marrow, they do go out into the other lymphatic tissues like lymph nodes, plasma cells, tonsils, etc. after being processed into plasma cells in response to antigens. The T cells from stem cell from the thymus marrow, they come to thymus and they become immunocompetent T cells, which again circulates through the lymphoid tissue and the organs, and eventually the plasma cells also recirculate to the bone marrow again. So basically they keep traveling between the immune organs. So far I have mentioned to you all the immune organs, they keep traveling. Now after the immune organs I have finished, I will go to immune cells. Now cells of the immune system all originate from the hematopoietic stem cells, which is self-renewing, and it goes to the myeloid progenitor cells in the bone marrow, as well as the lymphoid progenitor cells are there. The myeloid progenitor cells are responsible for growing into dendritic cells. Also they grow into granulocyte monocyte progenitors, developing into monocyte macrophage lineage or neutrophil, or they become eosinophilic progenitor cells and grow into eosinophil. They become basophil progenitors growing into basophils and mast cells, and also they can uh, uh, they are also precursors of megakaryocytes and erythroid progenitors growing into erythrocytes and platelets. On the other hand, the lymphoid progenitor cells can in the peripheral tissue grow into natural killer cell. They can also become the T cell progenitors growing into Th or T helper cell or T cytotoxic cells, as well as they become B cells, and in the peripheral tissue they become mature B cells, as I have mentioned to you. And also the lymphoid progenitors also can form dendritic cells. So dendritic cells can be lymphoid derived as well as myeloid derived dendritic cells. Neutrophils, as you know, 50 to 70% of white cells, 7 to 10 hours they last in blood, then migrate to tissues. The first responders of any immune response often, and they are motile and phagocytic. They respond by leukocytosis in infection, and they are important for neutrophil extracellular traps, as you know, very important for autoimmune diseases like lupus and some vasculitic conditions. The monocytes and mononucleus cells circulate in blood for 8 hours around and mature into macrophages or dendritic cells or tissue macrophages. They can digest and or present antigen. Uh, they also have surface receptors for antibodies or opsonized antigens, can be its function. Dendritic cells can be heterogeneous, as I have told you, myeloid or lymphoid origin. Antigen presenting cells presenting to naive T cells is one of its function to function as a APC. It also actively extends and retracts sampling antigens, and almost like amoeba it can move around, and it captures antigen in one place and then migrate to present antigen in another place, like in lymph nodes, as you've seen that the antigen presentation and B cell maturation happens there. So when the bacteria goes under the skin, you have this dendritic cells which release cytokines to activate neutrophils, macrophages. They also produce chemokines to act on monocytes to produce further macrophages, and these macrophages and dendritic cells they eventually go into the lymph nodes where they present antigens to the T cells to produce immune response. T cells also have T cell receptors equivalent to immunoglobulins on B cells. TCRs can only recognize antigen with associated when associated with MHC glycoproteins. Now T helper cells, CD4 cells, usually they uh, they uh, they respond to antigen presented by MHC class 2, and cytotoxic T cells respond to MHC class 1 presented antigens. Once T cell binds to antigen, it triggers cell division to form both memory T cells and effector T cells. Now a little bit about the gamma delta T cells and alpha beta T cells. Predominantly almost all the T cells are alpha beta T cells belonging to CD4 and CD8 cells predominantly, but there is a small percentage of T cells which are gamma delta T cells, and 60% of them do not have CD4 or CD8. They are negative for both, double negative, and only 30% have CD8 positivity, and they have no MHC restriction unlike alpha beta T cells. However, they represent only about 1 to 10% of cells. They are important in certain infections and autoimmune diseases. Actually, interestingly, in utero, it was the gamma delta thymocytes are more predominant, but from about 18 to 20 weeks during gestation, the gamma delta T cells decline and the alpha beta T cells keep increasing, and they become the predominant cells at birth, and gamma delta cells become negligible. T helper subsets can grow, or naive CD4 T cells can grow into one of these 5 different types. It can become Th1 cell under the influence and cell signaling through this pathway of these cytokines and the cell signaling, and the Th1 cells produce IFN, TNF and IL-12 and promote autoimmunity. They can also, under the influence of IL-4 and 6, become Th2 cells and produce IL-3, 4, 5, 9, 13 and promote allergy and also parasitic infection related response. It can become Th17 cells through the influence of IL-1, 6, IL-23 and TGF beta, and Th17 produce IL-17A, IL-17F, IL-21 and 22, and they promote autoimmunity and inflammation. And under the influence of IL-6 and activation of the BCL6 gene, they can become T follicular helper cell, produce IL-21, IFN gamma and TNF alpha, and helps for follicular zone B cells. And under the influence of TGF beta and FOXP3, they can also grow into T regulatory cells, and they produce TGF beta, IL-10 and CTLA4, and they cause immunosuppression. They suppress any immune response and may actually enhance sometimes infection, but they keep a check and balance on developing autoimmunity and aggressive immune response. So this is very important picture to remember. T cell differentiation also has 2 other important facts. Naive T cells are activated only by dendritic cells, whereas memory T cells are activated by macrophages, dendritic cells as well as B cells. So what happens is, once they are activated, IL-2 is produced, which is also known as T cell growth factor, and it eventually divides 2 to 3 times per day for 4 to 5 days after an antigenic stimulus, and it grows into a population of memory and effector T cells. So this is how antigen driven T cell differentiation happen. On the other hand, B cells are formed and matured in bone marrow. They exhibit antibody receptor on membrane. They once naive B cells bind antigen, they divide rapidly to produce plasma cells or effector B cells or memory cells. So this is a plasma cell, this is an electron microscopic picture. NK cells are activated by viral or tumor antigen that are expressed on affected cells. They recognize altered activating receptors and inhibitory receptors. They release preformed effector proteins from secretory granules known as granzymes and perforins, and they can kill altered cells by inducing apoptosis and antibody dependent cellular cytotoxicity. This is a picture of an NK cell. So these are the 3 different types of cytotoxic cells. One is CTL, or typically CD8 positive T cells. Other is NK cells. They both have almost similar effector molecules. Perforins and granzymes are important effector molecules. Interferon gamma, TNF Fas, and Fas ligand. So, and they also release these granules, perforins and granzymes, and directly can kill cells, and also they cause fast ligand interaction. The similar mechanism by CD8 positive T cells and NK cells. Whereas NK T cells produce effector molecules include for them IFN gamma, IL-4, GM-CSF, IL-2 and TNF, and they act via mainly fast ligand interaction predominantly and can activate NK cell indirectly also. So this is a graft versus host disease situation. This is a graft versus tumor situation, where the cytotoxic T cells get activated through the recipient non-transformed cell by MHC TCR interaction by antigen presentation and fast ligand activation, and this eventually lead on to apoptosis or programmed cell death. On the other hand, if a graft versus tumor disease where the cytotoxic T cells are activated via interferon receptor activation pathway and TCR MHC class 1 activation pathway, leading on to release of perforins and granzymes I have mentioned to you, and they create holes in the cells, and the cells get transformed and undergo apoptosis or cell death. So this is a very important picture called immunological synapse. This is the first one. On the left side is a T helper cell and antigen presenting cell synapse. On the right side is the T helper cell and the T cytotoxic cells and the target cell, which I mentioned to you, in that case maybe viral or tumor related cells. So if you see both the pictures, you can see almost everything is similar, except that in T helper cell the antigen processed into peptide is presented through MHC class 2 under the influence of CD4 to the TCR on the T helper cell, and there is also CD2 LFA1 LFA3 and LFA1 ICAM1 interaction, CD45R CD22 interaction, and CD28 B7 interaction, also known as co-stimulatory signals. This is very important because without the co-stimulatory signals the T cells cannot get activated even if all the others are active. If you compare these two cells, what is the difference you can see here? The only difference here is, here in cytotoxic T cells the antigen processed into peptides are presented through class 1 MHC, unlike it is in class 2 MHC here, under the influence of CD8, and you can see that there is no co-stimulatory or CD28 B7 co-stimulatory interaction is not there in this case, because you know the cytotoxic T cells, just a few minutes back I mentioned how they act by different mechanism. So all the other things are similar, except that the cytotoxic T cells do not have co-stimulatory signals. So if you see here again how the antigen, external antigen through class 2 MHC is presented by the dendritic cells to T cell receptors on T cell, and depending on the various types of stimulus, it can become either Th2 produce IL4, or it can become Th1 produce IFN gamma, and that needs IL12 and IL18 as I mentioned in one of the preceding pictures, or under the influence of TGF beta, IL6 or IL23, it can go, the resting T cells, helper T cell can go to Th17 pathway. I've also mentioned to you the other pathways previously, like T regulatory cells, etc. So this is the very important picture, the last few pictures, which shows how the antigen presenting cells activate T cells. However, as I mentioned to you, if by some reason the B7, which is one of the co-stimulatory molecules is absent, there will be anergy, there will be no activation of T cells. Anergy means no response. And if there is also blockage of the CD28 by antibodies, again in spite of B7 being present, there will be no activation, there will be anergy. Similarly, if there is no response between CD28 and B7, response is not there by some other mechanisms, or by giving molecules like abatacept, CTLA4, which can also block and cause no response. On the other hand, if this everything is intact in a normal pathway, any immune response activating T cells produce IL2 or the T cell growth factor and give rise to an activity. So you can see how important is co-stimulatory signal. This is more powerful than TCR and the antigen response by MHC. Now I've talked to you about immune organs, immune cells. Now I'll talk about the immune molecules. The most important effector molecules include antibodies, complement, cytokines and interferons. Antibodies are also called immunoglobulin. They belong to gamma globulin fraction of serum protein in electrophoresis. Y-shaped or T-shaped polypeptides, as you can see, you all know from your undergraduate knowledge. There are 2 identical heavy chains and 2 identical light chains, and there are 5 isotypes IgG, M, A, D and E. How the antibodies or immunoglobulin function in killing bacteria? Number 1, it can activate the other further B lymphocytes. Antibodies can produce further it can activate B lymphocytes in response to binding to the bacteria and producing further plasma cells and secrete more antibodies and producing memory cells. It can also act as opsonins like saponification binding to bacteria, which enables or enhances phagocytosis. It can also cause antigen clumping and inactivation of bacterial toxin like you can see here, and this also helps in the process of enhanced phagocytosis. The antibodies along with NK cells and eosinophils can also activate, as I mentioned, antibody dependent cellular cytotoxicity. That can also be there. The bacteria, antibody and the NK cells together act in that. So this is a unique immune response called ADCC. And then you can also have triggering of mast cells degranulation, releasing products of mast cells which can kill bacteria, and it can finally activate complements and give rise to again the destruction of the bacteria. The complement system we have classically three pathways: alternate pathway, classic pathway and lectin binding pathway. All of them act on C3 getting converted into C3b and C3a, and C3a is inflammatogenic and produces inflammation. C3b eventually is deposited on the microbes and opsonization happens and it causes phagocytosis, and this can act on C5 dividing into C5b and C5a. C5a produces again inflammation like C3a, and finally this activates the membrane attack complex or C5 to C9, causing lysis of microbes as the last step. So the same thing in this picture, whether it is a classic pathway by microbe binding with antibody or immunoglobulin, or alternate pathway by the MBL, alternate pathway, or by the lectin pathway through MBL, all of them act on C3, producing C3a and C3b. C3a cause inflammation, and C3b along with once you get opsonization with the bacteria or the microorganism, it again helps in cleaving C5 into C5a and b. So C5a again produces inflammation like C3a, and C5b helps in triggering membrane attack complex formation causing lysis of bacteria. So I have revised the grossly the complement activation system by these two slides. Cytokines are the low molecular weight proteins. They bind receptors, alters gene expression, and they can produce cytokines and cause biological effects on the target cells. They have autocrine action, that is acting on the cell itself. It has got paracrine action, is the surrounding the cell, or endocrine action in the distant action by bloodstream. The molecule can have actions. They are also in other organs far away from the origin site. The cytokines regulate immune responses, and it is pleiotropic in action. It has got different effect on different cells. So if you have to remember in cytokine, these two factors are very important. One is it can be autocrine, it can be paracrine, it can be endocrine, and it is also pleiotropic. Very important property of cytokines. Now interferons are very important. They are group of signaling proteins. There is type 1, which include alpha and beta, type 2 include gamma, type 3 include lambda. They have got the ability to interfere with viral replication and also inhibit tumor growth. They activate immune cells such as natural killer cells and macrophages, as well as they enhance T cell activity. So predominantly the type 1 and type 3 interferons are important for antiviral immunity, and type 2 interferon is important for antimycobacterial immunity. The broadly the induction of type 1 interferon by primary induction and secondary induction. The primary induction is by cell intrinsic inducers of interferon, which include the RNA triggers and DNA triggers, which are fragments of this, and viruses and the virally modified cells. Cell extrinsic inducers of interferon one include free chromatin, neutrophil derived mitochondria, neutrophil derived neutrophil extracellular trap, and apoptotic or necrotic cell debris, and nucleic acid containing immune complexes. All these things can act on on the cells through TLR7 and 9. They can cause secondary induction of type 1 interferon producing interferon alpha, whereas the cell intrinsic inducers act on the cells through REG1, MDA5 and C-gas and produce interferon beta. So these are the two broad mechanisms of type 1 interferon induction. Effector immune functions. I've talked about immune organs, immune cells, immune molecules. Now I'll talk about immune mechanisms of the innate immune responses and other immune responses broadly in the last part of the talk. There are normally, apart from the well-designed immune system, there are other barriers which prevent or protect us from infection and invasion. So epithelial cells in the intestine and respiratory tract, the physical barriers like tight junctions between the cells, mucins which are heavily glycosylated, they prevent attachment of microorganisms. Defensins are peptidic components and they can cause direct killing of microorganisms. TLR5 present on the basolateral surface and only invading bacteria, not the luminal commensals normally present in our gut, can cause TLR5 signaling. TLR5 signaling can activate the innate immune response, however, and also the adaptive. However, the TLR5 is do not get activated by commensals because our body and the GIT and respiratory tract has got whole lot of microorganisms. Our microbiota are needed for our normal homeostasis, so they don't trigger unnecessary immune response. So they are very specific. They will respond only to invading unfamiliar bacteria. Intraepithelial T lymphocytes also can recognize bacterial glycolipids. So these are normal barrier functions before the immune system come into play. So broadly, innate and adaptive immune response is, I will tell you in a very simplistic way, the innate immune response is like a reflex. It's like how the baby responds. If the baby is hungry, thirsty or upset, it will cry. Or sometimes, for example, someone is trying to put a hand across your eyes, you may immediately try to close your eyes. So this is a reflex response. So we have some fixed pattern in our genome which can remember few hundred patterns of microbes and their proteins and their products like lipopolysaccharide, lipoteichoic acid, glycans, peptidoglycans. Moment these are there, the immune system will blindly respond. If I see that, I will attack immediately. So that is innate immune response. It is very perfect and it is immediate and rapid and very robust. So it takes a very short time and it releases all kinds of superoxide, nitric oxide and cytokines, killing or neutralizing these harmful products of the microorganisms. On the other hand, adaptive immune system is more precise. It is imperfect, but it is slow and it needs a gene rearrangement. It is clonally decided and it has to respond specifically to the invading or the encountered antigen specifically. So that's how the B cells and T cell act, and they are less robust but little bit more specific, but they are imperfect. So it is like an adult who say, give me a chance, I will learn more. Give me a chance, I will learn more. This is the language of the adaptive immune system. Adaptive immune system says, give me a chance, I will learn more, so that it can produce memory cells, protect from future attacks, etc. Whereas innate immune response is immediate, robust, it is perfect, but it only can act against few hundred things, whereas adaptive immune system can modulate and change the type of immune response, either to cause clonal expansion or clonal energy of specific T and B lymphocytes depending on the invading or the encountered antigen. So pattern recognition receptors are therefore pathogen associated molecular patterns and damage or danger associated molecular patterns. This is very important. There are different types of pattern recognition receptors for innate immunity. The secreted PRRs, there are endocytic PRRs, there are signaling PRRs. The secreted ones act by complement activation and opsonization, whereas endocytic and signaling PRRs act by uptake by phagocytes or by inducible innate immunity. So you can see the pathogen can induce either soluble PRRs, which can cause either protease cascade or opsonization, which causes degradation, or they may produce the microbial antigen through the MHC to T cells, or it may be internalizing PRRs, which can be internalized, and even again through cytosolic PRRs it may cause gene expression causing cytokine and attacking the microbes, or it may be by signaling PRRs through TLRs or toll-like receptors, again causing the gene expression for cytokine production, etc. So there are different ways that the PRRs or pattern recognition receptors act. The pathogen recognition methods by PRRs broadly are three mechanisms. One is called non-opsonic recognition. So you can see there are membrane PRRs which can bind to this is a pathogen, bind to the pathogen. And there are serum opsonins. So opsonins, if they recognize so the opsonic receptors, then there can be opsonic recognition. So a pathogen may be recognized by a non-opsonic pathway or opsonic pathway, but there can be multiple recognitions also. You can have both opsonic recognition like this, as well as non-opsonic recognition like this in this. And eventually what that does it cause? It again produces TLR and produces inflammatory response. It can cause phagosomal recognition and microbial killing, or it can cause NLR or inflammasome protein related response, giving rise to again microbial killing. So and ultimate end result is inflammation and microbial killing. So interaction of antigen presenting cells with T cells, again you can see the PAMs act through lipopolysaccharides in the bacteria, and they may be endocytosed, and antigen can be processed through MHC to T cells and producing cytokines which act on bacteria to kill them, or it may be through the signaling PRRs which can like TLR4 pathway, and it causes upregulation of NF-kappa B, producing pro-inflammatory cytokines again. Ultimate end result is production of pro-inflammatory cytokines to attack the bacteria invaded cells. Now induction of inflammation happens by TLR signaling using by IL-6, TNF alpha, various chemokines and addition molecules. Internalizing PRRs, as I have mentioned to you, by ingestion, phagocytosis, by upregulating NADPH oxidases, phagosomal membrane oxidative burst, and superoxide ion production killing microbes. Or it may be inducible nitric oxide synthase pathway causing reactive nitrogen intermediate production, nitric oxide and peroxynitrite production, which kills bacteria. The NLR inflammasome, IL-1 beta and IL-18 also through the pathway can be bactericidal, and it may be uncontrolled with multiple pathways giving rise to endotoxic shock. So the inflammation can be controlled, it may become very uncontrolled, and it is vigorously activated multiple pathways. Activation of adaptive immunity, however, is different. The antigen presenting cells at the site of inflammation, they mature, and the maturation happens by upregulation of chemokine receptors. And they go to the lymph nodes and activate T cells. They internalize by internalizing pattern recognition receptors, they help in antigen presentation. By signaling PRRs, as I mentioned, they enhance co-stimulatory molecule expression like CD80 and CD86, and they can also cause TLR ligand activation. The TLR ligands can activate antigen presenting cells and thereby produce TH1, TH17 inducing cytokines. So these are various TLR molecules, very important, out of which TLR 3, 4 and 7, 8, 9 are very, very important. And TLR 3 is predominantly only endosomal compartment, and the recognized pathogen is usually dsRNA, which may be a product of a viral or endogenous product. Finally, the last two slides. The type of lymphocytes can be B cells, helper T cells, cytotoxic T cells, regulatory T cells, natural killer cells. B cells on antigen exposure, antigen on pathogens and soluble antigens, they cause production of antibodies, and they neutralize the pathogens, they cause phagocytosis, they cause complement activation. These are their effective functions. Whereas effector functions. Whereas the helper T cells, their antigen recognition is by antigen is presented by professional antigen presenting cells to them, and they secrete cytokines, and their effector function include activation of excuse me, macrophages, activation of other T and B cells, and causing inflammation. Cytotoxic T cells, the antigen recognition involves antigen presented by infected, by especially virally infected cells and malignant cells, and their effector function include elimination of the infected or malignant cells, and in that process sometimes they themselves also die out along with the target cells. Regulatory T cells, they do not have any antigen recognition function. They actually regulate or suppress the immune response. So they keep a checks and balances on the B cells, helper T cells, cytotoxic T cells indirectly. And NK cells are a specialized kind of cytotoxic T cells. Their antigen recognition again depends on self antigen, foreign antigens on host cells, which include viral or sometimes tumor cells, and their effector function include elimination of infected and malignant cells. And finally, the take home message is the primary immune organs like thymus and bone marrow, secondary immune organs like spleen, lymph node and MALT, have cellular components like neutrophils, basophils, eosinophils, mast cells, monocytes, dendritic cells, T cells, B cells, NK cells. Through their effector molecules like immunoglobulins, cytokines, interferons, complement activation, they cause pathogen clearance. They may lead on to autoimmunity if it is unregulated. They may cause autoinflammatory response by NLRP or the inflammasome pathway. They may cause allergy by Th2 pathway. They may cause immune deficiency if they are absent, either T cell arm or B cell arm or both, or the complement of the phagocyte arm, or they can cause cancer if there is abnormal proliferation. And autoimmunity when the self versus non-self differentiation cannot be, is it lost by the T cells which was educated in thymic education early in life, if that is lost. These are 3 YouTube videos I recommend you to see. The first one is somewhat like what I have spoken. The second one is for laymen, for public, to explain the normal immune function of the immune organs. And the last one is a very interesting, you must see, it's called 2-photon microscopy. So you can actually see live inside in vivo in animals and vessels how the T cells or cytotoxic T cells and B cells, antigen presenting cells, they act with each other. Really live tagged cells by 2-photon microscopy. You can see it is about 2 minutes or 3 minutes video, very interesting. Thank you for your patience. Thank you very much.